Welcome to LongGame Ventures' first portfolio demo day. I'm Will Harborne, and we're an early-stage venture fund investing at pre-seed, seed, and Series A in longevity biotech. We focus on frontier technologies, leveraging the latest advances in therapeutic drug discovery, enabling tools and infrastructure for drug delivery, advanced bioengineering, and computational biotech and AI. We see ourselves as signal investors, meaning we conduct deep due diligence on both the science to ensure the companies we back target the root causes of aging, and the business and founder aspects to de-risk and identify the world's highest potential teams with the biggest visions, extending maximum healthy human lifespan. We believe 2026 will be pivotal for longevity biotech. Aging and longevity are exploding as topics. Global demographics are shifting rapidly. By 2030, 1 in 6 people will be over 60, fueling massive demand for preventative and personalized medicine to address age-related disease. At the same time, convergence in AI, genomics, and broader biotech is enabling scalable interventions that could add decades to healthy human lifespan, developed faster and cheaper than they were a decade ago. This is driven by AI integration, e.g. AlphaFold accelerating protein modeling, new in vitro models like organoids, and automation advancing that shift work away from expensive, slow animal and human labs. These factors, combined with global competition from China's advancing biotech ecosystem, are accelerating regulatory evolution. Examples include the FDA's April 2025 roadmap to reducing animal testing, which prioritizes modern techniques like AI models, organoids, and organ chips for IND and preclinical safety in biologics. Longevity biotech is the leading edge of innovation and the field with the greatest potential impact, where these technologies and regulatory unlocks will compound fastest. We've made 15 investments to date, with our portfolio companies raising over $100 million in follow-on funding from fantastic investors. We've built a network of later-stage investors, both generalist funds and biotech funds and family offices, who rely on our signal in the longevity space. We met with hundreds of founders, selecting only the most promising. Today, you're going to hear from 13 of them, all at early stages, tackling themes like longevity gene therapies, mitochondrial rejuvenation, scalable, high-quality stem cell production, and more. These founders have massive visions, but they're also building scalable technology with real potential to push human lifespan boundaries. And most importantly, they're all backed by rigorous science that we vetted, from peer-reviewed breakthroughs to founder-led teams with PhDs at top institutions. So before we get going, some quick logistics on this event. We'll kick off with 5-minute video pitches from each company. After which, each of my co-partners, Manish and Sebastian, will provide a brief commentary on why we invested in the massive upside we see, drawing from our due diligence on the tech scalability and market fit. We'll intersperse Q&A, so use the Q&A chat within the public chat channel, and your questions will get read out and answered by the founders. The whole event should wrap in under 2 hours, with recordings uploaded post-event. One last thing, if you're an investor and something you see today resonates, please reach out. We're here to facilitate intros and co-investment opportunities. You can also follow LongGame Ventures on LinkedIn for updates and email team@longgame.vc to schedule a deeper dive. Now, on to our first portfolio spotlight, ImmuneBridge.
Hi, my name is Nina Horowitz, and I'm the CEO of ImmuneBridge. I'm here to tell you about how we're manufacturing the future of cell therapy. We are a small startup based in San Francisco, California. We have expertise across machine learning model development, bioprocess engineering, and cell therapy design. And we're backed by some really visionary investors, including NFX, Insight Partners, Ventures, which is European Merck, and of course, LongGame. Cell therapy uses cells as living drugs to attack diseases inside the body. So the scans I'm showing you here are from a woman named Lori. She was dying from lymphoma. She had failed 6 prior lines of therapies. Nothing was working for her. And then she received a single dose of a CAR T cell. And within a month, her scans had entirely cleared. She has never received any treatment since then, and she's still healthy to this day, 7 years later. The picture on the top right is us together at the Boston Public Library just last year. Data like Lori's triggered a Gartner hype cycle for cell therapy. So people rushed into the space. They were founding companies left and right. These companies were being acquired for up to $12 billion. And then everybody realized that manufacturing cells is actually really hard, and the field contracted. And in 2025, we've been in an IPO drought. But if ImmuneBridge can solve the manufacturing challenges, we're going to usher in a new era where countless new curative therapies are being developed for all kinds of diseases. Why am I so obsessed with the manufacturing? Well, here's what it looks like right now. 20% of patients are dying while they wait for their therapy to be manufactured, and the cost per patient is between $500,000 and $1.25 million. With the ImmuneBridge platform, these numbers go to 0% and just $10,000. The other challenge with cell therapy manufacturing is finding good cells. So cells vary really significantly from one donor to the next. I'm showing you here clinical data from a trial where patients all received cells from different donors. As you can see, the patients who received cells from good donors, the blue line, had a 14 times higher likelihood of progression-free survival than the patients who received cells from suboptimal donors. So of course, the big question here is how can we predict this in advance to make sure that every patient gets a good quality therapy? The ImmuneBridge platform solves all of these issues. So we source and screen from those donors that are actually capable of driving a cure, and then we turn them into consistent, affordable, and large-scale therapies. The way we do this is through three key technologies. The first is that we've developed machine learning models that can predict donor performance. The second is that we have designed a proprietary small molecule called IBR403 that lets us expand immune stem cells up to 100,000-fold in culture while maintaining their multipotency. And the third is that we've designed processes to be used in bioreactors for both this stem cell expansion and the downstream immune cell differentiation. All of these processes are GMP ready and ultra low cost. And what this means is that we're going to have both superior clinical results and superior market access because we're putting out a higher quality product at a much lower cost and a much larger scale. In terms of competitive landscape, there's really nobody who can do it the way we can. There's plenty of companies working with immune cells or induced pluripotent stem cells, but each of these comes with their own unique challenges. We are the only company capable of integrating high throughput donor screening with stem cell expansion as a large-scale, reliable source for manufacturing. We've spent a lot of time optimizing the use of IBR403, proving its capability to develop multiple different immune cell lineages downstream, working on our engineering processes, our bioreactor processes, and now we're finally ready to start a phase 1/2 clinical trial. So we're raising $50 million, which will take us through our tech transfer, our clinical manufacturing, and the trial itself, and will also enable us to develop a bunch of additional assets through partnerships. So while internally our focus is on NK cells for cancer, we will also be developing red blood cell, neutrophil, macrophage, and even mitochondria products for rejuvenation in aging populations. And because we can make any immune cell therapy or any immune cell byproduct, we are going to guarantee ourselves revenue across diverse markets. And this is a really exciting time in the cell therapy space because it used to be that cell therapies were only used for people who were dying of cancer. But now we're seeing cells used for people with Alzheimer's disease or liver fibrosis. And who even knows what else might be possible with cells if you could manufacture them at large scale and high quality. So I want to leave you all with a quote that I love. It's from the first liver transplant doctor, Dr. Thomas Startzel. And he said, "The history of medicine tells us that what was inconceivable yesterday and barely achievable today often becomes routine tomorrow. I want cell therapies to be that routine intervention that's available for anyone who needs them, and ImmuneBridge is going to be the company that manufactures that future." Thank you so much for your time, and I'm happy to take any questions.
And congrats on your board member becoming the director of ARPA-H, which is a $2 billion a year breakthrough moonshot bio funding entity of the US government. So with that, we love that you guys have a broad enabling technology for the entire cell therapy space. Could you elaborate on why cord blood cells are superior to, say, PBMC derived or iPSC derived?
Absolutely, yeah. So first of all, thank you so much for your support and for giving me the opportunity. I think the important thing about using cord blood versus these other sources actually has part to do with the biology of cord blood itself and part to do with regulatory. So on the regulatory side, cord blood comes from babies who were just born. This is actually already an FDA licensed therapeutic. These babies don't have any viruses. It's very easy to be sure that you're getting a high quality starting material in terms of contaminants. Biologically, there are even more benefits. So these babies are 0 days old. They have long telomeres, they have healthy mitochondria. It's a really clean starting population that has massive proliferative potential as well as downstream functional potential. So I refer to cord blood as liquid gold. I think it's a great thing for any cell therapy to be using.
For sure. Yeah, it's also a good source of young MSCs.
Yes,
To get them
absolutely.
injected for joint injuries and other applications. So
Yeah.
second question, then I'll open it up to the audience. The secret sauce is this HSC expansion method you have, achieving 100,000 fold expansion. They can allow you to produce many other cell types beyond NK cells. T cells have been hot in recent years. Can you just elaborate on the advantages of NK cells versus T cells, and then other cell types that you're excited about in the future?
Yeah, absolutely. So I will say that I do love T cells. T cells are not in our pipeline. That's because I think we can really uniquely able cell types that other people have been struggling with. So natural killer cells are great because they can sense virally infected cells, stressed cells, senescent cells, and potentially cancerous cells in a very heterogeneous manner. So you don't need a single antigen to target. So I think they're really great as potentially a senolytic, an infection preventer, and things like that. The reason that we then have these other cell types in our pipeline, red blood cells, mitochondria is just an organelle actually, and macrophages, is that I want to move beyond just the oncology space to the longevity space. And we're seeing a lot of really promising early advances in things like fibrosis and other areas, but with the same manufacturing challenges that have plagued natural killer cells. So I think T cells are doing just fine. That field is going to chug along on its own, but I think we really have the opportunity to help advance these other cell types into the clinic. And that's where I think we can have the biggest impact.
Cool. All right. So opening it up to questions, we have from Chris Bradley applauding the presentation here, and Greg Timblin asking about the MOA of the small molecule.
That is a secret, unfortunately. I can't say that on a webinar, but if you want to diligence us, I'd be happy to open up our data room.
Cool.
Amazing. Thanks very much, Nina. So next up, we're now moving over to Andy at Vincere Bio.
Vincere Biosciences is developing USP30 inhibitors that have potential for Parkinson's disease and other age-related indications. Traditional medicine has a problem whereby diseases are treated separately, but we all know that aging is the biggest risk factor for most diseases. Mitochondrial dysfunction is a particularly compelling hallmark of aging that contributes to Parkinson's disease, heart failure, and kidney disease. When mitochondria are damaged and dysfunctional, they can cause a cascade of problems, including inflammation and immune activation, that can contribute to aging and cell death. Our USP30 inhibitors enhance the natural process of mitophagy that bundles up damaged mitochondria and returns the cells back to a healthy state. We have a pipeline of molecules that are nearing clinical trials led by brain-penetrant, orally bioavailable selective inhibitors for Parkinson's disease, some topically optimized molecules that were recently partnered with Beiersdorf for anti-aging skincare, and peripherally restricted molecules that have potential for cardiorenal indications. Parkinson's disease is particularly compelling because mutations in this pathway cause early onset familial forms of Parkinson's. Recently, animal data has backed up this approach, showing that if you knock out USP30, you can prevent neurodegeneration and reduce the alpha-synuclein accumulation in the animal model of Parkinson's. Our molecules have been shown to very robustly increase the mitophagy process in the brains of mice, and excitingly, they do so more robustly in aged mice, where we know that mice have a deficiency in this process, a deficiency which is also shared with aging humans. The molecules have also shown protection in multiple forms of acute kidney injury, actually being able to restore kidney function when delivered after the injury has already occurred. We think this opens up a large set of additional commercial opportunities, but also I think is exciting from a geroprotective perspective, where this shared mechanism of aging shows protection across multiple therapeutic areas. Our company is founder-led and backed by some great world-class advisors, and we've been able to recruit a phenomenal team of drug developers who have taken drugs not only to the clinic, but all the way through to market approval and understand what it takes to be successful in this space. 2025 has been a very exciting year, starting with a out-licensing for anti-aging skincare to Beiersdorf, one of the top skincare companies in the world, as well as a financing that gave us the capital to move our lead molecule for Parkinson's through IND enabling and CMC to get ready for clinical trials. And then finally, a recent grant of $5 million from the Michael J. Fox Foundation that gives us some great scientific validation, but also additional capital to de-risk the program. The coming year has very clear objectives. We're going to wrap up the GLP tox studies and prepare for clinic entry and have a solid phase 1 plan to demonstrate safety, pharmacokinetics, and target engagement, including testing in some Parkinson's patients. Now, this IND stage, phase 1, is very common point of partnering for CNS assets, typically at a pre-IND or post-phase 1. And we have some great conversations undergoing with pharmas who are very keen to develop in this space. We're hopeful that we'll be able to get a transaction done over this timeline that will bring the right partner on to conduct the longer Parkinson's disease trials. And to strengthen position in that conversation, we are looking to raise some additional capital now that would give us the ability to run the phase 1 ourselves and starting conversations with potential lead investors. And we anticipate that this will just be 1 or 2 new investors joining our cohort to get this through the next stage. With that, we'd be happy to chat if this aligns with your strategy.
Thank you, Andy, for such a great presentation. Yeah, so pretty exciting. Mitochondrial dysfunction is kind of a causal hallmark of aging. So, I mean, despite all of this thing, there's a lot of translational gap. Nothing has moved into clinics. I mean, despite one of the very early FDA approved. I think I do want to highlight one of the very interesting things which we see in Vincere's approach is a solid biology around target engagement and lead molecules. And it's very rare. I've worked in this field and to identify molecules which can selectively clear up damaged mitochondria, because that's a distinction here. So very interesting, and congratulations on the recent Michael J. Fox grant. Maybe what I would like to hear your perspective on the recent approvals. And overall, I see this as a positive for the entire field in general, but what's your perspective in terms of like how do you differentiate compared to the other competitions which have moved into clinics?
I guess I can take that one. So I heard somebody actually say the other day that rising tides raise all boats, and I liked the way that that was said. And that's really how we think about the competition. As you said, the mitochondrial space has been a challenging one. And there's been a lot of question of can you even target mitochondria safely, because they are such key players in just your general cell health, right? They're essential. So seeing the first approval by Stealth for a rare indication, that's gives us great confidence that mitochondria can be targeted. And then other competitors that may be a little bit ahead of us give us insights on what to avoid, what traps are out there, which we've learned a tremendous amount from these guys. And then of course we're fully differentiated. We are actually one of 2 companies that are targeting USP30, the only non-covalent modifier that doesn't bind to everything kind of cysteine rich non-selectively. And so we're in a really good place from a differentiation perspective, and we're actually targeting, the machinery that degrades the damaged mitochondria. So we're very, very specific, leaving the healthy mitochondria alone. So I think overall, from a competition perspective, we're really happy that there is competition. If you have a good idea, there should be competition, and we're happy that they're succeeding because their success is ultimately our success.
Yeah, no, absolutely. I see Chris and saying the good job, Andy, and Nathan. Congratulating you guys for progress. So that's exciting. And maybe one more thing which I could ask is like there has been recent data around USP30 and which was a main profile publication in Science, which talks about conceptually moves the field forward in terms of mitochondrial DNA mutations and USP30 mediated mitophagy being involved in that thing. So maybe if you can briefly comment for the audience that what does it mean for the field?
Yeah, absolutely. This was really nice work by Patrick Chinnery's group that you're talking about, and reduction of heteroplasmy, right? And that's an area that we've known about from in the longevity field, trying to reduce heteroplasmy, and they were able to show independently of us that USP30 is involved in this, and by reducing USP30, you're able to reduce heteroplasmy. So we're pretty excited to see more coming out of that front. You know, we've one challenge of this being so important in so many indications is that we can't look at all of them. And it's really fantastic that some of these like really stellar academic groups are doing some of this work basically for the field. And yeah, absolutely. So excited to see that.
Next up, we're going to go over to Jonathan at Herasight for a very different strategy on how to impact lifespan and health span.
Yeah, so I'm Jonathan from Herasight. We do genetic testing of embryos, and you know, IVF has been around for a long time. It's about 50 years old now. And in the standard process, you stimulate the ovaries, produce some eggs, and create embryos from eggs and sperm. And so, you know, in the past, people have selected pretty arbitrarily between which embryo to implant. What we do is we reveal a lot more information about the embryos. And for about 30 years, you could test for those really rare single gene disorders, the kind that cause Tay-Sachs or sickle cell anemia. But the vast majority of what we care about, whether it's disease traits or height or intelligence, is highly polygenic, meaning they involve thousands, tens of thousands of genetic variants that add up to an overall probability that you'll develop type 1 diabetes, be a certain height, et cetera. So that's the kind of thing we do. And these are some of the traits we offer, physical and mental traits, whether it's selecting against schizophrenia, bipolar disorder, selecting in favor of height, intelligence, and so on. And we have an overall longevity index that I won't show on the slides, but that sums up the overall sets of risks from different diseases, and we put those into disease-adjusted life years. So you can also kind of compress all the information we give into an overall longevity index. And this is how we compare. There are a few other companies in the space, but this is how we compare with our best competitor, which is Orchid. And that is to say, when you look at the overall R squared values, that is the amount that we can predict genetically of any particular trait, we beat them on every trait that we offer, as well as offering more traits than any other company offers. And that includes intelligence. So just to take an example, we've developed the world's best IQ predictor. This has been 4 years in the making. And if you have 10 embryos, we can predict a spread of about 15 IQ points. That's actually half of the overall variance. And it's remarkable, but if you were to have 10 siblings as adults, or you had 10 embryos, the actual difference in range between IQ, between the lowest and highest scoring, would be about 30 points. And we can now predict just over half of that. Okay, and if you want to know what is the appetite for this sort of thing, well, among Americans, almost half say they would already select for cognitive ability. In Singapore, the number is almost 80%. So in those countries where this isn't taboo, nearly everyone wants it. And even in the countries where there are some taboos around it, about half the population want it. And that's, again, just selection for IQ, separately from disease traits. Next. And what we have, probably the most important thing that we can do is actually not just the quality of our predictors or the quantity, but we have an entirely new way of doing things that allows us to scale internationally. And we serve people around the world right now. And that is because there are two ways of doing this. To reveal all these traits about embryos, you can either whole genome sequence them directly, which is very hard and very expensive. Orchid is the first to do it. We can do it too. But we actually developed an algorithm where we can take data from a common test that's done in any IVF clinic, that is the test for Down syndrome, and we can take the really sparse genetic data from that, and then we can whole genome sequence the parents, knowing that embryos are a function of parental DNA. And this is a sophisticated way of doing things, but we do both short and long read sequencing. And from that data from the parents, plus the data from the Downs test, we can fill in the rest of the genome of each discrete embryo and thereby reveal the entire genetic sequence of each discrete embryo without directly whole genome sequencing it. And what that allows us to do then is to send people to almost any IVF clinic in the world of their choice. They could go to Dubai, where Sebastian is right now, or Mongolia, and they can take this Downs test, request the raw data from that test, then we sequence the parents, and we put that together to reveal all this other information. And I'll try to be quick about this, so I'll finish in a minute or two because I don't have too much time here. But what this shows you on the right is what our interactive report looks like on the left. That's for any given trait. Since we can infer or impute the whole genome of each discrete embryo, and we have these really cool predictors that are quite powerful, we can reveal all of these interesting things to people who either have a family history of disease on the one hand, maybe it's schizophrenia, maybe it's Crohn's disease. And overall, because everyone faces some risks, we can compare the embryos on the right-hand side. You can see that against one another with respect to all of the traits that we can predict. So we've already served about 100 people and have about a dozen babies born. And basically in the future, and the future is now, it's basically this year, we're going to go from serving predominantly Americans and a few Europeans to serving the whole world. And we have a few ways of doing that. One way is to get government contracts with countries in the Middle East where cousin marriage is common, and we have a solution to that. Another way is to get insurance subsidies, but we're also working directly with IVF clinics and direct to consumer, because with this new imputation strategy, we can actually go on podcasts like I do all the time and just capture people's attention, say, this is what we do, this is interesting. Go to your IVF clinic, request your raw genetic data from this common aneuploidy test, and we can do the rest for you. Next. And that's pretty much it. So we're going to go from 3% of Western nations use IVF right now. About half of our customers already do it electively in order to optimize traits or reduce disease. And we're going to go from 3% to a much, much bigger percent in the coming years. So thanks.
Great. Thanks, Jonathan. I firmly believe that embryo selection is going to be the greatest public health advance of our lifetimes, probably, and it's low-hanging fruit. I mean, we already have the information, right? So we might as well take advantage of it. So, yeah, I think we don't have that much time. Maybe one quick point would be, you've got the backing and blessing of a lot of the most famous Silicon Valley entrepreneurs and prominent people, and you're speaking with some governments as well. What would be the argument to governments to avail themselves of this technology?
Well, the argument to governments in the Middle East is because cousin marriage has been so common for 1,000 years that there are a lot of genetic problems that could be easily solved by just choosing the embryo with the lowest runs of homozygosity, basically meaning it's just going to have fewer diseases and inadvertently have a higher IQ without even testing for IQ, simply because there are 1,000 ways to screw up the genome. And when you marry a cousin, those ways can manifest themselves. And there are a few ways to improve it. So that's the one pitch for governments. But the other pitch is places like Israel, where I just was, and Singapore. They already have robust public health systems and mandatory genetic testing in some cases. They certainly subsidize it already for everyone else. And so it would be a really short step for them to also subsidize not just single gene disorders, which is a tiny fraction of them, but polygenic disorders, which is most of what people care about when it comes to diseases, but also other traits that really have public health benefits, including, I think, intelligence. And I'll just say, sorry, quickly, 10-second version of that is, intelligence is correlated with a host of important life outcomes ranging from length of life to subjective quality of life, and these are measurable, to just overall good health outcomes, precisely because intelligence is correlated with long-term planning, self-control, and a variety of other traits that are indirectly connected with health traits. So thanks, Sebastian.
Excellent. Amen to that. And everybody should check out Jonathan's book, Creating Future People. It's online, in which he goes into the ethics and long-term considerations of embryo selection.
Thank you very much, Jonathan. Going on next, we've got a presentation from Ryan at Syntax.
Hello, everybody. My name is Ryan Clark. I'm the founder and CTO of Syntax Bio. So we have invented a cell programming technology that we are using to realize the potential of stem cells for cell replacement therapies. So the concept of this type of cell therapy is to take stem cells, which have the ability to be converted into any cell type, and to take them and to convert them into a tissue-specific cell type, and to take those tissue-specific cells, transplant them into a patient's diseased or damaged tissue. And the idea here is that that transplant will be a single dose that will cure the patient of that ailment. Because stem cells have the ability to be converted into any cell type, this modality has a lot of promise because very, very large indications can be treated if you can make the right cell type. And pharma has been investing in this space for about 20 years now, and there's been a lot of exciting progress where recent clinical trials have shown that, for example, a dose of pancreatic cells can cure a diabetic patient, making it so they don't need an insulin injection ever again, or paralysis can be cured with spinal neurons. So the sector has been growing slowly but surely since the derivation of induced pluripotent stem cells back in 2006. So there are over 100 clinical trials now across many different tissue types and indications. And there is a lot of deal value associated with each of these assets, with close to $500 million in biobucks on average for just a single asset. And pharma has been striking a lot of these deals over the last several years. But what you probably noticed with this graph showing clinical trials over time is that this curve starts to plateau in the recent years. And that is because the manufacturing is a massive challenge. Manufacturing a single batch of cells typically takes 1 month or many months. There is a high risk of variability that can lead to batch failures. And at the end of the day, a dose of cells will cost millions of dollars, essentially making this modality commercially not viable. The reason that the manufacturing is so difficult is because it's analogous to where the punch card era was back in the 1950s, or where the computing era was back in the 1950s, where we were using punch cards. So it works to compute, but it's very slow, it's very manual, and it's hard to QC. So how this works with the current method is that scientists will take their bioreactor growing their stem cells, and they sequentially add different recombinant proteins over a many months procedure. And so if you mess that up, you have to start over. So the iteration cycle time is very, very slow as you're optimizing your protocol. And again, it's very onerous and it is hard to QC. So what we are doing at Syntax is we are bringing this modality into the modern era, analogous to how microprocessors and software are used for computing. And we're doing this through our technology, which is called cellular algorithms or cellgorithms. So cellgorithms are encoded in the language of the cell, which is DNA. And what they specifically encode is a CRISPR system that we invented. And so how the process works is you deliver your cellgorithm just one time to your stem cells, and it runs pre-programmed gene regulations that turn genes on or off in a stepwise manner. And this is automatic, and it's very, very fast. So it's just like pressing run on a computer program, and the lines of code are executed over a matter of days instead of months. So what this physically looks like in practice is just a single tube of cellgorithmic DNA is used to drive the state changes required for the stem cell going to the tissue-specific cell type. So this is the actual example of our control method for differentiating pancreatic into pancreatic beta cells. So you can see it's dozens of inputs. And again, we reduce this down to just one input to drive the changes. So we have shown that we can take a stem cell, program it down all three germ layers. We can make cells of the eye, cells of the muscle system, of cartilage, of the hematopoietic system, and what we're focused on today is the pancreatic system. Quantitatively, what this looks like is our process takes about 1 to 2 weeks on average for every lineage that we have tried this on. Again, we are reducing down to this number from many months long process for some cell types. And we streamline this down to just 1 media or 2 media formulations required. What this ultimately leads to is up to a 20-fold reduction in costs for some of the most difficult lineages. So 2026 is going to be an exciting year for us. We just recently received a grant from the Breakthrough Type 1 Diabetes Foundation to fund our pancreatic beta cell work into diabetic animal studies. And we have just filed some IP on our 4th generation cellgorithm system that is capable of sequential repression and activation. Up to 10 different programs can be encoded in a single cellgorithm. And one of the most important things that we're working on is a ecosystem of partnerships. So rather than making therapeutics ourselves, we are embedding ourselves into the ecosystem to work with the clinical transplant expertise, the clinical scale manufacturing partners, as well as the inputs who make GMP DNA and different types of media formulations. So keep your eyes peeled for some announcements that we'll be making in the coming months with some investment and partnerships with some of these large organizations. On the financing front, we have just closed our Series A in Q4 of last year. We were excited for this to be led by Astellas and Illumina Ventures, and very excited to have LongGame participating in this alongside Draper Associates in one of the final closings. So thank you all for your support. And what we're working on now is a top off of this Series A that will be closing in the coming months. So interested in talking to folks for that as well. Thank you for your attention, and looking forward to meeting some of you sometime soon.
Fantastic. Yeah, I think we haven't got Ryan here for Q&A, but yeah, Sebastian, maybe you want to go ahead anyway.
Sure. Yeah, I'll just sing their praises a little bit here. So obviously, Ryan and team are kick-ass founders. They have a very prestigious cap table. Astellas, the #1 regenerative medicine pharma Japanese company, is a partner, an investor. Illumina, the largest genome sequencing company in the world. DCVC, Civilization Ventures Portal, etc. And I don't know if they're announcing this yet, but they have a collaboration with, shall we say, the #1 hospital in the world, hospital brand in the world. You might guess who that is, and an investment from them. And so, one interesting fact about them is that with Astellas, they were working on a project that took several months, 6 months to make this cell type. And Syntex was able to get it down to just a couple of weeks, which is unheard of. So, we like this technology because it is a broad enabling technology for every single type of cell therapy. And we would love for some of our portfolio companies to collaborate with each other, ImmuneBridge and Syntex and a handful of others. And I think there's synergy there. So anyway, yeah, we're happy to connect you with the team if anyone wants to chat.
I think it's people will notice there are a lot of companies in our portfolio related to the stem cell supply chain and manufacturing process. How do we make these cells scalable and usable in the clinic? And Syntex fits that really well, but also then fits very nicely in terms of generating revenues early through partnerships and licensing, rather than kind of having that sort of binary option of developing specific therapeutics and cell types themselves. So anyway, on that note, we'll go on to our next presentation, also related to the cell therapy space, with Ohad from Renewal.
Hello, everyone. Thank you, LongGame, for inviting us to present in this demo day and for your continued support. My name is Ohad Gafni. I'm the co-founder and CSO of Renewal Bio, and today I'm going to share with you how we're renewing human health. I would like to start with this painting of Lucas Cranach the Elder, the Fountain of Youth. It shows elderly, weak people and sick coming from one side of the fountain and coming out young, healthy, and ready to party. Unfortunately, we haven't found this fountain of youth yet, but we're making stride towards alternative. And there are quite a lot of evidence in the literature where blood from a young mouse was transfused into an old mouse through a blood circulation, shared blood circulation, and the young blood managed to rejuvenate the immune system, but not only the immune system, also the brain, the heart, and other tissues. And this set of experiments actually showed us that transplantation of young genetically matched cells and tissues can rejuvenate tissues and organs. And of course, a great promise to obtain young cells for various therapies came with the first derivation of embryonic stem cells. And for those who don't know, embryonic stem cells are the group of cells that later differentiate to any cell of your body and form the entire embryo. But there is a major roadblock mimicking the complexity of the bioenvironment in differentiating these cells. And that's why we created Renewal Bio and to basically address the problem to obtain young cells and tissues for transplantation. And this technology is based on an evolution of discoveries from Jacob Hanna's lab at the Weizmann Institute in Israel. And we've created a new entity of therapy we coined as Stembroids, which are human stem cell derived embryo models or developmental models from cells alone. So no eggs, no sperm, no fertilization, allowing authentic holistic differentiation to various cell types with just one platform. And this technology featured on the covers of Nature and Cell, the most prestigious science journals. And many in the scientific community believe that this discovery is actually a Nobel Prize worthy. And today I want to share with you a little bit on our tech and its market. So I want to start with our incubator, our roller culture that we've developed, which closely mimics the uterus environment, allowing embryos to grow outside of the uterus. This was a very complex goal, of course, because the conditions in the bioenvironments are very tricky to mimic. And when we took a mouse that was fertilized inside a mouse naturally, and we took the embryo to grow in our incubator, we saw that it develops properly, giving rise to all cell types, all tissues, all organs, as they normally happen in uterus. The development of this device allowed us to support proper embryo development outside of the uterus for the first time. And in this movie, you can see the gastrulation process. This is the most important process of differentiation, generating the various cell types and tissues that make organs. And as Louis Wolpert said, it is not birth, marriage, or death, but gastrulation, which is truly the most important time of your life. You can see here from these images that the embryo not only growing in size inside our incubator, but actually properly develops and creates the axis of development. You can see the head, the tail, the heart beating, the liver, and a lot of other organs, and just developing just like in utero. And this slide is a little bit scientific, but please bear with me because it encapsulates the whole process. So we start with embryonic stem cells or any other stem cells, and we transition them into our naive conditions. From these naive conditions, we can not only make the embryo proper cells, but we can make also the supporting environment of the extraembryonic tissues like the placenta, the umbilical cord, and the yolk sac. And when we aggregate them together in a special plate, we form these developmental models termed stembroids. And the models develop proper structure mimicking human embryonic development, as you can compare between the textbook image on the right here and stembroids on the 8th day of the protocol. You can see that they very closely resemble, and the stembroid actually mimicking the development. And the beautiful thing about our platform is as we develop further and as we move forward with development, we can make different cell types, and we can basically isolate different cell types for different indication. And the only thing that differs in the platform is the way we isolate the cells. As we move forward with development, we unlock more and more cell types that we can use for different indication, and then later on tissues and organs that we can use. And this is an immense opportunity because the stembroid platform is super valuable because it helps us understand what's biologically possible. And then we can decide what makes sense clinically and economically. And our technology revenue potential is immense, of course. $117 billion only for the cell therapy market. But of course, it can be used as a progenitor cell source, a platform for drug testing, and a lot more. Thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Ohad. So yeah, this is definitely a very unique company in our portfolio, and kind of falls into what we would class as a category of kind of cell and tissue replacement. It's a little bit different than, instead of trying to slow down and reverse damage and aging in old cells and tissues, actually looking to replace them with new young cells and tissues. But of course, the challenge then is, how do you get those young cells and tissues? And that's what Renewal is really solving. So yeah, I think from our perspective, and fantastic scientific team, great investors and vision, and tackling that kind of challenge of how do you at scale make these cells. I know that, congratulations, since we invested, you've closed your latest sort of $5 million round, and have the firepower now to kind of move towards commercialization. So we'd be keen to hear more about how you're approaching that next step, especially the first cell type, HSCs. What does it look like technically and commercially actually to make those next steps?
Sure. Thank you, Will. So first of all, it was the seed round and it was 15 million. We just closed it. That was very exciting in this, unbelievable landscape. So our first milestone is, of course, unlocking day 28 to 30, which captures actually a few cell types. But as you mentioned, hematopoietic stem cells are the first cells that we're going to deal with. We chose the plastic anemia indications as bone marrow failure disorders. But as I mentioned in the talk, the hematopoietic stem cells are a great way to rejuvenate all tissues, especially the blood, of course, and, dealing with pathogens and, just rejuvenating the immune system and not only the blood. So, of course, that's our first goal, as you mentioned. But the beauty about our platform is that, we're get to day 28 and 30, and that's what we're doing right now. We're actually unlocking the different stages, and we're understanding that there are a lot more cells that we can actually use. We found out, about a few months ago that endothelial cells are they have a market for that as well, for vascularization. So we're actively testing, and we're seeing that these cells are actually functional and they're forming tubes.
Yeah, I think one of the things, from an investor perspective that makes it really exciting, as you highlighted, is that even a single cell type, has a massive market potential here. How do you kind of view that trade-off internally around continuing to progress the platform and go to kind of later and later stages and unlock more cell types versus focusing on commercialization of the existing cell types?
Yeah, that's a great question. It's a question that we get quite a lot, but our focus is mainly about the platform and moving it forward. So we know we specialize in pluripotent stem cells and developmental, trajectories. So we know what we know, and we've collaborated with the right people like ImmuneGene and other partners that help us take our hematopoietic stem cell product further. And of course, for the other cell type, within endothelial cells, we have a great partner here in Israel, and we have a lot of different partners for different cell types. So yeah, that's how we're moving it forward to get the experts from every field.
Fantastic. Thanks so much, Ohad. So next up, we're going
Thank
over
you.
to the world of gene therapies with Matt from Oisin Bio.
Aging is the largest unaddressed therapeutic market in history. And I don't think it's possible to meaningfully extend human lifespan with pills. We need more powerful tools. We need genetic medicines. And for the first time in history, we actually have them. I'm Matt Schultz, and I have an unusual background. I actually started in cybersecurity. I got into biotech with the thesis that the essence of life is information. And if you can manipulate that information, you can do anything. Today, I run Oisin Biotechnologies. We're building genetic medicines for health and longevity, therapies that the FDA will approve and that pharma will license. I previously founded ImmuSoft, initially backed by Peter Thiel and Tim Draper, where I developed the first engineered B-cell therapy to ever enter human trials. That was before there were any FDA-approved gene or cell therapies at all. And it worked, but it took over a decade to go from idea into the vein of a patient. That experience shaped how we built Oisin. This team has taken two entirely new classes of therapies from discovery to the clinic and through pharma licensing. We know exactly where these kinds of programs can die, and more importantly, how to get them across the line. The longevity dividend is real. The value of slowing aging in the US, even by a modest amount, is worth tens of trillions of dollars. But aging proper is not regulated as a disease. So this huge opportunity exists, but only if you can approach it correctly. Now, for the longest of time, this was for practical purposes impossible. But recently, two monumental shifts changed the game entirely. First was the rise of the GLP-1 drugs. What was once just an obscure metabolic therapy became the most valuable class of drugs in history. And more importantly, they really demonstrated how much patients are willing to pay out of pocket to look and feel better. Second, the use of genetic medicines expanded dramatically in the early 2020s. Regulators, industry, and the public became comfortable with genetic interventions at a scale that no one saw coming. This combination has made longevity medicines feasible for the first time ever. Oisin's strategy is simple. We start with indications the FDA understands that allow us to directly attack the biology of aging. We're starting with frailty because physical strength is everything as you age. Loss of strength drives the loss of independence, injury, hospitalization, and even mortality. Fortunately, muscle biology is well understood, and the pathways that regulate skeletal muscle growth are some of the most de-risked in medicine. There are literally decades of human data targeting this pathway. The failures have almost always been around manufacturing and delivery. And we think we can succeed here where the others have failed. After just a single injection of our muscle building therapy, the animals were 50% larger and twice as strong. Our first indication is buttocks augmentation, clear endpoints, cash pay. And we get paid to validate a frailty therapy in humans. If we clear that bar, moving into aging proper is actually pretty straightforward. Our second program selectively deletes adipocytes without collateral damage. Think of Botox for fat. This work attracted AbbVie, the undisputed world leader in injectable aesthetic medicine. AbbVie led our Series A after paying roughly $2 billion for their last attempt at an injectable therapy for body sculpting. The delivery has been the Achilles heel of genetic medicine ever since it existed, and it's been a fundamental roadblock for longevity companies. Viral systems don't scale, they can't be redosed, and they have limited payloads. Our platform combines viral efficiency with the non-viral safety and scalability attributes. So delivery is our moat, and it's what pharma actually buys. Our Series A was led by AbbVie Ventures. We've already completed our milestones. This capital advances 2 assets, muscle and fat, to IND. These are exactly the kind of programs large pharma companies license for billions of dollars. If you're interested in longevity, built like a real biotech, I'd love to talk. Thank you.
Okay, cool. So yeah, this is a very exciting technology that addresses the biggest problem in gene therapy, which is the delivery problem. So backed by AbbVie Ventures, AbbVie is one of the largest pharmas in America. They partnered with Google's longevity company, Calico, to the tune of $3 billion. And they are very keen on longevity. And so happy to have them as partners. You know, gene therapy is the modality that I would bet on that is most likely to not only slow aging, but reverse aging, to reverse the aging process. So, you know, rewriting the code of life, we could just deliver Yamanaka factors or factors, similar factors we discover in the future that could actually turn the clock back rather than slowing the progression. Another fun fact is that there are companies who shall go unnamed that are offering gene therapies, for example, follistatin, one of the genes in the Oisin pipeline. They're offering sort of a bootleg version offshore. People are reporting that it benefits them, that it works, that they have added muscle. I will personally wait to take Oisin's GMP grade, pharma grade version in the next couple of years. But yeah, so we're very excited about this company. And yeah, I think we can move to the next presentation.
Yeah, well, just wanted to highlight also, so and Matt mentioned there that you know, now they're actually advancing this initially, especially on the muscle side, towards buttock augmentation. This is, you know, we invested in this looking at a potential use case for frailty and extending lifespan and healthy lifespan. But a theme we see across the longevity world and across many longevity therapeutics now is they have these potential aesthetic uses, whether that's through bigger muscles in all areas of your body, you know, better skin and other things. And so I think a theme we're seeing is when looking towards commercialization, many of these companies now may consider aesthetic endpoints as their initial way of going to market before expanding into broader aging-related use cases. But anyway, yeah, thanks very much, Sebastian, for the commentary there. And next up, we've got Martin Jacko from Aperture Therapeutics.
So at Aperture, we develop a new class of medicines for neurodegenerative diseases that mimic the effects of protective gene variants, what we call supervariants, that we find in individuals with exceptional resilience to disease. Our mission is to develop first-in-class therapeutics for the growing population of patients with age-related neurodegeneration that is spanning multiple conditions from some of the most severe, like ALS, to some of the most prevalent, like Alzheimer's disease. None of these diseases really have any therapeutics that would make a significant impact in the life of individuals and slow or halt the progression of the disease. And so our unique approach leverages the the fact that the global population is genetically extremely diverse, and this really serves as a natural experiment that helps us to understand why some people do have neurodegeneration and progress through it fast, and others are more protected. There are some great examples of genetic variation playing a role in genetic risk as well as resilience, like genetic variants in PCSK9 that are protected from cardiovascular disease, or CCR5 genetic variants that are protected from HIV infection. Our mission is to identify which genetic variants are the largest impact genetic variants that provide resilience from age-related neurodegeneration. To do that, we have compiled some of the largest databases of patients with genomic and clinical data that we're able to mine using machine learning approaches to pinpoint which genetic variants are associated with slower progression of disease and later stage of onset. After identifying these variants, we're able to develop molecules like antisense oligonucleotides or siRNA that modulate the activity of the genes to mimic the effect of the protective gene variants and can be applied to the broader patient population that don't have the genetic protection. Now, we're very excited to combine large amount of data generated by nonprofits and academics to generate one of the largest databases of genomic and clinical data for ALS that helped us to identify more than 12 variants in different genes that seem to be protective from ALS. One of them is in a gene called CD33 that has shown where partial loss of function has shown a delayed disease onset and more than doubling of survival of patients with ALS. Now, we're very excited to see this because if anybody was able to develop a drug mimicking the effect of this genetic variant, they would have the best drug that ever existed for ALS, giving the patients with the disease multiple years of additional survival at least. So we have 3 programs ongoing for different genetic targets and neurodegenerative diseases. The one for CD33 is the most advanced. We have recently announced a development candidate that is now entering IND-enabling studies, and we're raising 10 to 15 million in additional capital to progress 2 of those programs towards clinical trials at the end of 2027. Our second program is in discovery stage. As I mentioned, most of these therapeutics are antisense oligonucleotides or siRNA. We're also developing and have IP for a delivery technology that actually allows bringing these molecules into a specific cell types where they are relevant, and very excited to develop a second generation of the drugs that would have further improved pharmacology. And so our first indication that we plan to enter is ALS. This is a high unmet need condition that represents a very significant market of more than 10 billion globally, and more than half of that would be expected to be in the US. And so we are on track to be in clinic, as I mentioned, at the end of 2027. So that is everything from me. We will be very excited to talk more with you and tell you more about the programs that we're developing and the data behind them. We believe that these are some of the best targets and novel targets for neurodegenerative diseases that haven't been previously drugged, and that based on the human genetic data would have a massive impact on the progression of the disease. And if you feel like it, please reach out to us. You can also find us on our website, aperture.tx.com. Thanks for your attention. Hey, everybody. Great to be here.
Cool. Thanks, Martin, for the great overview. And maybe I can just briefly talk about what we like about Aperture. I think they have one of the very solid scientific background in terms of utilizing a human genetic data, and specifically for the human genetic resilience data. I mean, historically, if you see, the neurodegeneration field has seen a lot of setbacks, and most of this is partly because of not the very potent preclinical models. So guiding the approach, which is based on the human genetic data, I think is the best logical and rational approach. And congratulations, Martin, for nominating your DC candidate. Very exciting there. Maybe I would like you to elaborate, and I think which is a very important piece here is the magnitude of effect you see with this target. So this is amazing because I mean, currently as you see, the standard of care for ALS, the current are within 3 or 5 months, not even more. So this is dramatic if it works, even partially. So maybe for our audience, if you can elaborate a bit on that.
Sure. So yeah, as I mentioned, in ALS, the typical survival is 2 to 3 years, and so having effects or targets that show 50% inhibition, giving patients 2 to 3 years is massive. As you mentioned correctly, the current therapies give typically 2 to 3 months of extension of survival, and you really have to squint your eyes when you look at the clinical trials to actually believe even that data. To some, it actually might be disappointing, right? That we're not curing the disease, and 2 to 3 years might actually not seem as much. I think the general understanding is that, you will actually need more than one target to be modulated to actually cure the disease. And so we were excited to build a pipeline around multiple targets and actually expect some synergies between the targets as well. And so we're sort of excited to push these programs alongside and expect that we'll be actually built, by combining these therapies, we can potentially get to cure eventually.
Yeah, very exciting. And maybe one more thing I would like you to highlight on, like, how do you see these therapies expanding beyond just cognitive decline? I know there is a data and kind of MOA around these molecules. It's like working on inflammation and brain inflammation. There is certain data around them regulating systemic lifespan. Maybe you can better highlight on that. How do you see this broadening beyond just brain cognitive decline?
So in ALS, what we actually track is the disease progression, motor decline, and survival. And so we see that for the targets that we identify, they're sort of broadly induced and cause pathology through inflammation in different parts of the CNS based on the specific disease. CD33 have been previously identified to be important in Alzheimer's. That's where it sort of causes the issues with the cognitive decline. But we expect that, it will be sort of induced in a couple of different conditions based on, wherever the sort of pathology initiates. And so really the therapies that we're developing are sort of disease modifying and sort of broadly effective on, a couple of different phenotypes, really depending on what disease you're getting.
Very cool. No, pretty exciting in that thing.
Yeah, fantastic. Yeah,
All
so
right.
next
Thanks,
up,
guys.
we've got Aaron from Revel. Thanks. Hi, everyone. My name is Aaron Cravens. I'm the CEO and co-founder of Revel Pharmaceuticals. My background is a PhD from Stanford University in bioengineering, followed by a few years in industry in the Bay Area at Google X and other startups. I now lead the team building the first platform to repair structural aging. What do I mean by structural aging? Well, as we age, damage accumulates in our bodies. This damage accumulates both in cells and outside of cells. While the majority of the longevity space is focused on cellular aging, trying to either keep cells young or kill off old cells, cells are actually only about 30% of the mass of our bodies. The other 70% is non-cellular. This 70% is the non-cellular structures like collagen, elastin, the extracellular matrix proteins that cells live within, and that give our bodies their structure. These non-cellular structural materials also undergo aging, and they age in the form of accumulation of glycation damage, oxidative damage, and buildup of crystals like uric acid crystals in gout and calcium crystals in cardiovascular disease. At Revel, we are focused on this non-cellular component of aging. How do we reverse crystals, reverse oxidative and glycation damage in order to restore structure of tissues to a more youthful and healthy state? The advantage to doing this is our targets are very clearly defined, and removal is easily measured. This enables rapid preclinical and clinical validation down the road. One of the first programs we're focused on, and I want to share briefly today, is our work on curing severe gout. So gout is a crystal disease. In gout, high levels of uric acid in the blood precipitate out into your joints, causing painful crystals that are inflammatory and also physically damage the joint tissue. In very severe gout, patients go on a drug called Krystexxa. Krystexxa is an enzyme that basically dissolves the crystals. It's a biweekly IV-infused drug. It has a very poor patient profile. It's a high burden on patients in order to receive this therapy. And so as a result, it only treats about 5,000 patients a year of the approximately 50 or 100,000 patients that could be eligible. Despite this, it's still a billion-dollar drug. It sells over 1.2 billion a year currently. Now, what we're doing at Revel is developing a curative treatment for severe gout that would truly be transformative for patients. Our solution to doing this is to develop a liver-directed gene therapy that basically turns liver cells into biofactories for secreting the therapeutic enzyme, effectively Krystexxa that's been re-engineered for stability and activity in this format. This is a huge market opportunity. We think we could treat 10 times more patients, and it allows us to prove out our platform in a crystal disorder that has an established mechanism of action and established market already. So we view this as a relatively low risk on the clinical side. The reason why no one has done this before is there's a significant challenge to treating patients with a one-and-done cure for severe gout. The natural uricases, or Krystexxa-like molecules, are limited by folding and immunogenicity problems. We've overcome this challenge with a proprietary variant discovery selection and screening platform. We employ basically a device that allows us to assess millions of protein variants simultaneously in order to sort through optimized enzymes that can enable a stabilized, immune-optimized, and secretion-optimized protein. We've established a very compelling proof of concept in this application for gout, and that is rapidly normalizing the serum uric acid levels in mice. So serum uric acid is the biomarker that's used in gout. You want your serum uric acid to be as low as possible. The graph I'm showing here on the left is the serum uric acid of a gout model of mice. The mice that are untreated with the black curve have high serum uric acid that's causing damage. They have gout. These mice do not live that long. Following a one-time treatment of our AAV gene therapy drug, we show that we can bring down serum uric acid to below clinically measurable levels and basically cure these animals with just one dose. This is a powerful proof of concept for what we're doing, and we believe we can rapidly prosecute this program towards IND. In the second quarter of 2026, we're looking at nominating a development candidate molecule. In the third quarter, we're preparing for our IND meeting with the FDA. And by middle of 2027, we intend to submit an IND along with preparation for first-in-human studies in severe refractory gout. While gout is our first low-risk target, we are really excited about scaling this platform from gout to systemic repair. We have many other targets that in the structural aging space are very important. Glycation changes, oxidative changes, things that we would like to reverse. A few of these I've shown here with the various drivers associated. We're currently working on advanced glycation end product reversal with a first indication in the diabetic retinopathy space. That program, we have some really exciting human tissue data where we've taken 70-year-old tissue from cadavers and shown we can reverse the levels of glycation damage back to that of a 40-year-old. That work was done in collaboration with a leading pharmaceutical company in the space. So we have some really compelling programs that we're working to prosecute. And it's quite an exciting time at the company. To date, we've raised a little over $16 million from investors and non-dilutive NIH grants. And we're just finishing up a bridge round to get us to the IND filing. With that, we're Revel Pharmaceuticals, and we are repairing damage that aging leaves behind. Thanks for your attention, and look forward to answering questions.
Nice. Thanks, Aaron, for that overview. Pretty interesting. Maybe I can just briefly talk about what we like about Revel. So I've been fortunate to work on AGEs during my tenure at the Buck Institute, and I think AGEs are one of the fundamental drivers of aging, even though they are very overlooked. There are very few companies, to our knowledge, which have been seriously tackling AGEs. So that's pretty interesting that you're doing something in this field. Very interesting. And for the gout program, it's very interesting. It's a hugely unmet need. There is a huge pharma interest. And for our audience who may be familiar, there have been recent deals in this space, which involves one of the Swedish biopharma company acquiring Arthrosi for $950 million, and Novo Nordisk's doing a Series A investments in a gout program. So very exciting in all this space. Maybe I would like to hear from you a little bit more in terms of like, how does the clinical trial of gout looks like? It's a very straightforward trial, which is something excites us as far as very early in the phase 1. You can get the indication signal, and sometime directly you can jump from phase 1 to phase 3 if the efficacy is pretty strong. If you can elaborate a bit more on that.
Yeah, that is something that's super exciting about gout and unique to gout. In the phase 1b studies, we can see the biomarker, right, is serum uric acid. As in the mice, within a few weeks, you have a very clear efficacy response as to whether there is a lowering effect with your drug. And the nice thing about gout is we all have actually high levels of serum uric acid. So with either healthy volunteers or patients, you can see a response. For AAV therapy, likely it will be with patients, right? But the response can come quickly, and you can see that, which is not the case, right, in many other indication areas. And as you mentioned, the gout space has become, it's very exciting. There's a lot of investment going in. Some recent acquisitions have occurred for small molecule oral drugs, and we're quite excited about that as a opportunity.
Yeah, no, definitely. And also maybe like broadly, like serum uric acid levels have been tied up with all-cause mortality. I think that is a very interesting point to note, because it just expands us beyond the gout. And I don't know, what do you think about that?
Yes, so high levels of serum uric acid are correlated with mortality. So there is part of our interest in gout as a crystal disease is this link to longevity, right? Same with calcium crystals. You get various forms of crystals and amyloids, and it's part of the pathological change and accumulation that builds up as we age. And the biomarker, lowering it as a biomarker too has longevity potential, even separate from just having gout. So I think that's an interesting expansion application.
Yeah. I see we have a question. I don't know if we have time for that, but a very quick question. Yeah, so we have a question from Ashish. He's asking, are you also working on other delivery mechanisms other than AAV?
We're discussing with another company on alternatives to AAV. We're not totally committed to AAV. The thing we like about it is there are some clinically approved vectors that are fully de-risked in human, and we know the liver targeting profile. They're very inexpensive to manufacture at low dose. AAV does have a reputation as being very expensive, but if the doses are lower, it's actually much less expensive. So we do have other things in the works beyond AAV, but right now that's the head of the pack.
Yeah, so switching gears a bit now, we're going towards a very early pre-seed stage company in synthetic biology, doing something pretty different than we've had so far. So handing over to Layla at Totality.
Hi, my name is Layla Strickland, and I'm a scientist turned entrepreneur and second-time founder who's been focused on the biotechnology of human milk since I struggled to breastfeed my own children. My kids are teenagers now, and over time, I've come to see human milk as a naturally evolved catalog of human health solutions that goes way beyond babies. Now I've teamed up with Patrick Shih from UC Berkeley to launch Totality Biosciences with a unique molecular farming platform that will functionalize the food supply with full-spectrum human milk oligosaccharides. Human milk contains numerous bioprotective components, and human milk oligosaccharides, or HMOs, are one of the most unique categories. HMOs support a healthy gut microbiome, they regulate the immune system, they block pathogens and reduce infection, and they contribute to even building a healthy brain. In the past decade, some HMOs have been industrially produced for premium infant formula, and research is ongoing to explore their benefits across a wide range of populations. But manufacturing challenges today limit their widespread use, leaving a significant market opportunity for those who can find solutions to today's particular hurdles. Today, HMOs are industrially produced using genetically engineered E. coli. After 10 years in the market, we now have a small industry with about 5,000 tons of production volume annually, maybe generating around $250 million in revenue. Behind the scene, there's actually a growing concern among both buyers and suppliers of HMOs that these ingredients won't reach their commercial potential because the products are still too limited and the ingredients are too expensive. Making HMOs in plants instead of bioreactors is a step change that leverages the natural metabolic engine of photosynthesis, resulting in low upstream costs. Meanwhile, the existence of processing infrastructure for food crops offers a range of downstream options that don't exist for HMOs produced in E. coli. Our foundational technology was developed in the lab of our co-founder and CSO, Patrick Shih at UC Berkeley. Patrick and his graduate student, Colin Barnum, created the world's first genetically engineered plants for HMO production by expressing specific enzymes for HMO synthesis in a model plant. Colin and Patrick proved that their plants can make a diverse array of HMOs, potentially even all 200 HMOs. They also showed that the plant-produced HMOs were similar to HMOs from breast milk for supporting the growth of gut-healthy bifidobacteria. And finally, they showed that HMOs could be made at least 10 times cheaper in plants compared with E. coli, setting the stage for the spin-out. The first goal of Totality is to take the genetic engineering from a research model into a food crop to create the world's first platform for photosynthetic milk oligosaccharides. As such, we're calling this platform FOMO, and our goal is to make sure that no one misses out on HMOs. The pre-seed strategy is focused on validation of two specific HMO targets that address the two prongs of our value proposition. So first, we want to make a cost-competitive version of a popular HMO ingredient today. We're focused on making 2-fucosyl lactose, or 2FL, at a cost that will expand its use for gut health across new consumer segments. Second, we want to make a complex HMOs that no one else can even produce at all today. So DSLNT is the perfect target for this. This is a life-saving molecule for preterm infants in the NICU, but it can't be scaled up today. Across infant and non-infant applications for these two programs alone, I estimate that this would be a $6 billion business in the US if it existed today. But of course, we're just getting started today. We're filling in our pre-seed round so that we can move quickly with full speed this year to deliver the world's first food crops for HMO production, backed by strong strategic partnerships that will set the stage for a focused launch and rapid growth. We're focused on key de-risking points, executing on a strategy to launch 2FL in 2030, followed by DSLNT in 2032. As the recurring revenues are established based on these two programs, we'll invest internally in new products and supply chain internalization, growing to become market leaders with 10 kiloton annual capacity and $2.2 billion in annual revenue within 10 years of launch. Patrick and I have been working side by side since the spring of 2025. With support from the LongGame and Replicator VC, we've been able to bring Colin on board. Our lab is up and running in Berkeley, California, and I've still got a little bit of room in the pre-seed round if you'd like to join us to functionalize the food supply with the world's first food crops for HMOs. Thank you.
Thanks so much, Leila. This is actually a company where we were the first check-in, and that's because of really Leila and Patrick making such a great founding team. Combination of sort of Patrick's expertise in synthetic biology with Leila as a second-time founder in the human milk space sort of really makes a very, very compelling case. I mean, what attracted to us to this, to Totality from a longevity perspective was, first of all, actually the focus on even infant formula as one route, making such a big impact on health span and lifespan. If you look at actually the difference between babies that are fully breastfed versus infant formula, there is big health outcomes for life, for your entire life, and all-cause mortality. But also HMOs potentially in adults can be critical for gut health, immune health, and so we see a really strong, interesting longevity angle here. Leila, we sort of obviously think about it a lot from the therapeutics angle, but of course you're also looking at it from the food angle, potentially replacing and adding many of these to infant formula. Could you sort of talk more about how those two parts complement each other?
Sure. Well, we always are kind of straddling this food as medicine and food and therapeutics, because we're working with bioactive components that have the potential for real efficacy. And so as a therapeutics approach or therapeutics play, we're about to become the only company in the world that has access to this complete library of biomolecules that have been through already millions of years of clinical trials in the 117 billion people who have existed on Earth thanks to these molecules. And so they have a very strong evolutionary history in supporting our survival and promoting our health that's well validated by our own existence. And so we're excited to be the first to be able to start to map those structures to functions and understand in much more depth than we do today how they're impacting mechanisms of inflammation and immunosenescence and some of the mechanisms specifically related to longevity. On the other hand, breast milk is very much a food for infants. And as a food, we know that it is safe. We know that we can be exposed to it without worrying tremendously about side effects and adverse events. And so from that standpoint as well, we think these are potentially some of the safest therapeutics you could ever have, and even safe enough when formulated properly to be used in the food supply. And we think it's important to intervene upstream of disease if we want to really maximize the lifespan and health span of humans around the world. And so it's kind of a two-pronged approach to human health. Let's figure out what the therapeutic potential is of this incredible class of molecules that we've never had access to for development. And then let's also find out what's the best way to use them to achieve the functional goals of making people healthier for as long as possible.
Thanks so much, Leila. Actually, one quick one. We've only got 30 seconds, but could you tell us why you chose DSLNT as as the first more complex HMO to go after, which isn't already widely used and widely available?
Yeah, so today around 10% of babies are born preterm, and around 10% of those are very likely to develop a devastating inflammatory disease of the gut called necrotizing enterocolitis. And DSLNT is a complex human milk oligosaccharides with no synthesis method today, but it has been produced at levels that have allowed preclinical work to be done. And so in rodent models of this necrotizing enterocolitis, we know that it improves the survival of these babies. And we also know that the pathology in the intestine with necrotizing enterocolitis is actually pretty similar to irritable bowel syndrome and other inflammatory disorders in the gut later in life. And so we think that if a scalable method of production were available, we could save the lives of babies in the NICU, as well as potentially have therapeutic efficacy in a much larger expanded population of people who are struggling with gastrointestinal inflammatory disease.
Thanks so much, Leila. So next up, we've got Robin at Deciduous.
Right. Good morning, good afternoon. My name is Robin Menzicani, CEO of Deciduous Therapeutics. I'm going to talk for a few minutes today about how we can restore the immune system's ability to remove senescent cells and how that leads to making a best-in-class therapy for lung fibrosis. A couple parts to my talk. First, I'm going to give you a primer on senescent cells and how they're causal to a number of different age-related diseases. Two, I'm going to talk about our lead compound, DCD80, which restores immune function in the lung and how we can position it to be best-in-class therapy. Three, I'm going to follow up on that and show you how DCD80 is useful not just in lung fibrosis, but also in other indications driven by senescence. And lastly, I'll give you a brief overview of the TPP and how we're moving this forward into the clinic. These are indications where senescent cells have been shown to be causal in human diseases, but also indications where removing them in preclinical studies has shown to be impactful in resolving those diseases. So just gives you a roadmap of where a good senescence approach can be applied. Okay. First model is in lung fibrosis. I'm going to explain the model and take you through the data. So in this model, we induce lung fibrosis via a chemical called bleomycin. Bleomycin induces fibrosis and senescence and inflammation in the lung. It happens quite acutely in the model. So by day 0, you're inducing. By day 7 to day 10, you start to reach the peak of disease in the animals. And so what we do is we treat once at the plateau of disease, which is day 10. We give one dose of this drug subcutaneously, and then we look at the impacts on fibrosis about 10 days post the therapy. So just to draw your attention, the black down here is the control animals. That's what healthy looks like. We looked at some cytokines related to inflammation first from the lung. So TNF-alpha, IL-17A are fibrotic cytokines. When they're elevated, you see more fibrosis. When they come down, you see less fibrosis. And so the objective is to reduce these. So we're showing with TNF-alpha, we can go back to homeostatic levels. IL-17A is also attenuated. But most importantly, we see a dramatic reduction in the fibrosis of these animals, looking at not just part of the lung, but the entire lung in totality. So about an 80% reduction of fibrosis in these studies. Visually, that's what it looks like. That's what an uninjured lung slice will look like. That's what healthy and good is. The middle is an injured lung that has fibrosis, so you can see the vast visual difference. But when we treat, you see a significant improvement just visually in these lung slices. Numerically, again, it tracks about an 80% improvement in the animals. We also looked at this from an Ashcroft perspective, which is sent out to a blinded pathologist. We don't tell them which samples are which. We just say, score these. And the higher the Ashcroft score, the more fibrosis. The lower the Ashcroft score, less fibrosis. And so left to right, healthy looks like this. That's what perfect would look like. A diseased lung with fibrosis looks like this. When we treat, we see a dramatic improvement, looking much closer to healthy than injured. Scoring-wise, 0 for the healthy control, so that's perfect, as I would say. 5 for the injured, so that's untreated, and then back to a 1 when we use this approach to treat these animals. So again, a dramatic improvement by the blinded pathologist. Okay. Most of the collagen 1A. Collagen 1A is a gene that is often overexpressed in lung IPF human patients, and so we wanted to see what would happen in our hands in preclinical studies if we could reduce this. And so it turns out this is what collagen 1A looks like at a healthy level. The higher it is, the worse it is. The worse it is in humans typically tracks with poor survival and poor respiratory function. That's why it's an interesting gene to look at. But with treatment, we see an improvement almost back to baseline. We also put this up against the best current drug in the market called nintedanib. Nintedanib was given this study 10 times versus 1 time of our drug. And we're just showing here that essentially nintedanib does not perform very well in fibrosis, which is well known in humans. But we see with 1 dose of our drug, we beat nintedanib even when given 10 times of the competing drug. So this is one of the reasons we think we could have a best-in-class therapy. If you look across the landscape and the research and literature, we can show head-to-head that we can beat most, if not all, the drugs that are currently approved for IPF. Okay. Second indication, so lung fibrosis is really interesting, but this is not just a single indication molecule. So multi-indication impact. And so just showing you a snapshot of some diabetes data. This one just shows that when we give 1 dose of this drug in a diabetes setting, in type 2 diabetes animals, we looked at A1C levels. A1C levels are very stable indicators of glycemic control over time. So they're very long-term indicators. Typically, the first study to get done in the clinic would be, if you're getting diagnosed with diabetes, is to look at your A1C levels and see if they're going up or down or maintaining. And so we should show in this study that even after we first put the animals on 4 months of high-fat diets, we then dose them once at day 120, and we show that the control obese animals tend to have increasing A1C levels without treatment. The ones we treat, though, go back to normal, healthy level controls from a single treatment over 2 months. So you get a durable effect for a few months from 1 treatment. Interesting for an IPF lung fibrosis trial because many, at least half of patients who have lung fibrosis also have type 2 diabetes. So you can actually look at both in a clinical setting. Okay, this is the TPP of the compound. So intermittent dosing, everything I showed you today was single dose, small molecule, so very low cost of goods, very good for patient exposure and convenience. Strong specificity of pathological senescent cells. You get really good on-target effects. You don't see any off-target effects, even at 30 times the effective dose. We believe this can be a therapeutic best-in-class for lung fibrosis and potentially other indications. The safety profile looks very clean in preclinical studies to date. The mechanism-based tox also looks quite clean. And most intriguingly, With these approaches, these are given systemically, and so you'll see a multi-indication impact, which I've shown you a snapshot of today, where we get both lung fibrosis improvements, but also diabetic improvements. That could extend into many more indications, as I could explain in the Q&A. So to summarize, failed immune clearance leads to the accumulation of senescent cells. The immune system, specifically in this case NKT cells, is what we activate with this molecule, can be re-stimulated to remove senescent cells. A single dose leads to expanding the NKT cells, removing senescent cells, improving fibrotic and metabolic disease. We don't see any serious adverse events, even up to 30 times the effective dose. The therapeutic window looks quite large. In the upcoming Series A, we will do is focus on testing DCD80 in lung fibrosis patients and associated comorbidities of lung fibrosis patients, which there are many. So sort of a basket study of aging if you zoom out a bit. Okay, thank you so much. Happy to take any questions.
Yeah, thanks, Robin, for such a great presentation. And I think like senescence in general is a pretty mature field in the aging biology, and one of the among the very early which went into clinic. The field has learned a lot from recent trials, so that's very exciting. And I do want to mention is like something which is very compelling for us in Deciduous is like IPF is a very hard indication. It's very difficult to target and everything. So seeing something with a single dose, such a dramatic effect, it's amazing, impressive, and that too beating a standard of care. So that's very exciting there. I think, yeah, in general, what I would be curious to learn how you're thinking of the clinical trials, because in the terms you have two programs which seems to be showing something very interesting result. Would love to, I know we have talked in past, you have some very interesting thoughts around that thing. If you can briefly speak about that, how you're thinking.
Yeah, absolutely. So, IPF is a orphan disease, not very well treated right now, very expensive. It's $200,000 per patient per year, current therapies. The thing about, I think most people don't appreciate about IPF is that there are actually multiple comorbidities commonly associated with that disease. So, for example, half of the patients have type 2 diabetes or are obese. Roughly the same number have cardiovascular disease, renal disease, kidney disease. 80% of them actually have gastric reflux. What's intriguing for us is because those indications are also senescence driven. And the reason we think we'll have an impact on those is not just from the diabetes data I showed, which there's more, but with our approach, it's systemic. So it's a subcutaneous injection. It goes to multiple organs. And so we're showing dramatic effects in the lung. We're showing dramatic effects in the diabetes case. We think there are also going to be effects on the cardiovascular, renal, and liver organs as well. And so you may see an improvement there. So it's just one of the things when you go into IPF, because of our approach, you can look at more than one single outcome. To collect data on other organs, it's a simple blood draw, an EKG, a urine test, et cetera. So these aren't complicated markers to pick up. And so what's nice about that from an investor perspective is, if IPF we think will look quite strong, but it also may look strong in other areas. We can pursue those afterwards as well.
Yeah, completely agree there. So I see we have a question on chat box from Flora. She's asking, is there a difference in the effect of aged NKT cells to be activated by DCD80 versus younger patients?
Yeah, it's a great question. So the question is, does it work in just young or can it work in older? So yes, we've gone up to 18-month-old mice, quite old, done IPF studies there as well. The effect looks quite similar to what I showed in this piece of data with the younger mice. Also, what I want to point out on top of that is, it's not just age that drives down NKT cell numbers. Even in younger animals who have diabetes, you see a suppression of the NKT cells, but we can still pick them back up, stimulate them and expand them even when they're suppressed, even at younger ages. So it's not even an age-related thing. It's actually disease and age together really suppress them. But the good news is with the right molecule, it can still activate them and expand them regardless of age or regardless of disease.
Nice. Maybe one next quick question is from Urmila. Nice work. Any thoughts about using DCD80 in liver, heart, or kidney fibrosis?
Yeah, it's one of the things we'll look at in that basket study for the phase 1A and 1B. To do it preclinically, I think was, it's not a focus area right now, but we do want to look at those markers in the clinical studies.
Cool. Thank you, Robin, for your time. And I think next up we have Julian from AdaptyvBio. So AdaptyvBio is working on a very interesting interface of AI and protein engineering. So,
perfect. Yeah, hey everyone. I'm Julian, co-founder and CEO of Adaptive, and we build automated labs to test AI-designed proteins. So there are now basically like dozens and dozens of AI models for protein design, and the bottleneck is really generating real-world data to validate which protein designs work and which ones don't, like kind of closing the loop between the computational prediction and the wet lab validation. And that's exactly what we're doing. So we've built a automated wet lab to actually make all of those proteins and test them for the different functions, process all the data, and then give our customers results with which they can improve their AI models. And we spend a lot of time automating a lot of the wet lab biology here. So this is very cheap and very fast compared to traditional CROs or compared to what people could do in-house. And also we build a lot of software around the wet lab so that our customers, the protein designers and the AI model builders, they can just go to our website, upload their proteins, get instant pricing, confirm their experiment, and then our wet lab workflows kick off immediately afterwards. And then basically you just get email updates whenever your experiments are finished, and you can download your data and then you can feed that into your AI models to improve them. And so this is a much nicer process than dealing with these sort of like CROs and sending PDFs back and forth and getting data in a PowerPoint and so on. Quick summary of traction. We raised an $8 million seed round at the beginning of 2025 where LongGame invested, and since then have been scaling quite nicely. So team is now 25 people. We tripled revenue in 2025, onboarded over 50 customers to our platform, and are working with some of the largest pharmas like Novo Nordisk, all the AI model builders like Chai, Boltz, Cradle, labs like Microsoft Research, dozens and dozens of tech bios that are designing thousands of novel proteins every month and testing them on our platform. So throughout 2025, we've really become the de facto data generation engine for protein designers. And now we're continuing to scale this in 2026 even more. So some plans here, what we're doing next is we're releasing a set of enzyme assay benchmarks. So protein designers can start designing new enzymes like PETase, so plastic degrading enzymes, and then test them on our platform. We've done all the wet lab setup for those, so we're rolling that out soon. And also a few months ago, we released a new platform called Protein Base, where we open source data. So we open source data for people to benchmark their models on and train new models with. And then those models, we again benchmark in our lab and release more data. So think of this as a mix of Hugging Face and GitHub for protein design. And on this platform, we're also running public protein design competitions where basically anyone can submit protein designs for a challenge. And then we test 1,000 or so for free in our lab and release that data. So we're creating like a sort of data flywheel with this. We're currently running one where we're testing binders against a Nipah virus, so one of the deadliest viruses and has huge pandemic risk potential. And we're generating new binders to block this. And the data is coming out next week. So stay tuned for this. It's looking really cool. We're just running all the assays here in the lab. Last thing I want to mention quickly is we're also releasing our API in the next couple of weeks. So we have had an internal API, but now we're rolling out public API access. And that means that basically anyone can integrate our wet lab with their protein design platform. And this is going to be cool because thinking a bit broader here, effectively, we think by the end of this year, we'll probably have like 95% of knowledge work, including science, effectively, being done by AI agents on behalf of companies. And so you can imagine these AI agents, they sit in the cloud and they design proteins and biological experiments, but they need the tools to interact with the real world. So if you think of Claude Code, for example, the big step change here was to have the agent live in your terminal on the computer where it has access to all the tools, like all your files, the browser, your email, and so on. And for science agents, the big step change will to give them access to experimental tools, like allow them to interact with the real world, to run real wet lab experiments. And we'll be the best platform for that, basically. So yeah, looking really forward to that. Check out AdaptyvBio.com if you want to test some proteins or if you know anyone who tests proteins.
Amazing. Yeah, thanks so much, Julian. Yeah, I think we love this as an infrastructure play, a bet on the fact that biologics and protein engineering is going to grow, it's going to continue to grow, particularly, looking at it from a longevity lens. We see that the growth, particularly around using protein-based modalities for targeting all sorts of longevity targets, is only going to continue to grow, beyond just small molecule approaches. And so that's very exciting for us. And also, this just represents a kind of democratization of drug development and the pieces that are involved. So you no longer need to have millions of dollars and a lab in order to make progress on the early stages of your drug development. You can be at home as an individual with a laptop, start to generate designs for the proteins that work on a specific target, and pay for the experiments to be run and get back data and continue to make progress. And that's huge in terms of accelerating timelines of getting new types of drugs and longevity therapeutics to market. I'd be keen to hear more from you around the kind of range of assays that you have in the wet lab and how that's growing and changing, and what you're seeing being demanded from clients now versus when you were first starting up.
Yeah, so what we started out with was basically binding assays, because, in the end, what proteins do, they just interact with other proteins, and that's like binding to other things, and that's what most interactions are being mediated by. So we started out with this, and it's still like 80% of what we're doing. It's really where like most of the markets are. This is antibodies, this is diagnostics, this is like all sorts of applications are basically binding proteins. And there's so many things to do where we're just starting glimpses of what happens when people design proteins that then bind to other proteins that are also engineered. Like there's kind of like sorry, am I lagging a bit right now?
It's fine.
Do you hear me, Will? Okay, sorry.
Yeah, fine.
So kind of like binding is still what most people are doing. Let me go on this slide maybe. And now up. Okay, I think it's lagging a bit less now. Okay, so binding is still what most people are interested in from a commercial point of view, but as the models progress in their capabilities, we think more and more molecular functions will be unlocked. So that's why we're already setting up the enzyme assays now, and we want to take it all the way to basically testing molecular machines on the platform. Right now, most people are interested in binding. That's what we've built out the infrastructure for. We're releasing the enzyme assays. We can do all sorts of developability parameters for therapeutic applications. Also, what we're really interested in is like more complicated functional assays. And so we're looking for protein designers that want to test those.
Thanks so much, Julian. So next up, moving back to small molecule approaches, introducing a very well-known company in the space, General Proximity, and Armand.
Hi, I'm Armand, and I'm the founder and CEO of General Proximity. General Proximity is solving the biggest challenge in drug discovery, which is that there are many well-known drug targets that we understand the biology of in disease, but where we lack the technology to effectively modulate that target in a human. The way this changes is that some new technology comes about and suddenly the undruggable becomes druggable. So we are developing a fundamentally new class of medicines that we call next-gen induced proximity therapeutics. At very high level, they work by inducing protein interactions or proximity events between two proteins in your body to drive some effect. Why is this so powerful? Essentially, all biological phenomena are downstream of binary protein interaction events. Meaning, any biological output you can think of is on some level controlled by two proteins interacting. And so if you can understand that interaction landscape, you suddenly gain the ability to control it and drive biology that isn't possible to achieve with any other modality. This is what we built our tech around. We pioneered this space. We built a set of platforms that allow us to understand for a given drug target which protein interaction events drive the biology that we care about. We've applied our technology to only the most high-value targets we could find beyond just having platform disease fit. Does the target make sense for our technology? The only other major criteria we use to pick targets is that do we think this program could eventually impact all humans on Earth? because of that, we've generally worked on transcription factors. Transcription factors are a class of proteins that are at the root of all health and disease. They are canonically hard to drug or undruggable, yet all transcription factors are controlled, even more so than most other proteins, by proximity events. A given transcription factor will have hundreds or thousands of protein interactions that orchestrate and tune every level of its function. So if you can understand the proximity landscape of a given transcription factor, you suddenly gain the ability to tune its biology in ways that you couldn't do before. Our flagship program is for the most important target in oncology, the master tumor suppressor in humans, a protein that controls all major tumor suppression pathways that's never been drugged. We have made, to the best of our knowledge, the most potent activator of this target ever created. We plan to put our first molecule from that program into humans next year. Beyond that, we've worked on a number of very interesting targets in neuro, cardiometabolic, and longevity. There are all sorts of very powerful outcomes you could get by modulating transcription in things like longevity. For example, mitochondrial biogenesis, cellular resilience, metabolic adaptation, autophagy, cellular reprogramming. All of these things are controlled by transcription. We've got a number of programs in this space that we're advancing towards the clinic as well. We've raised about $25 million to date from some of the best investors in Silicon Valley and beyond. We've received major grants from the National Cancer Institute and ARPA-H. And we've also just recently announced our first platform collaboration with a major pharma company, Daiichi. They're paying us to use our technology to go after some of their high-value internal targets. We're getting ready to raise our Series A in the very near future. If you'd like to learn more, you can check out General Proximity at prox.bio, prox.bio, or you can email me at armand@prox.bio. Thank you for listening, and thanks to the LongGame folks for putting this on.
So a couple comments there. Hey, well done. Very smooth, and you've been executing like a madman. So we're proud to be on the cap table. We have a broad audience on the call. So just by way of background, the induced proximity, pulling together proteins rather than the typical pharmacology of enzyme inhibition or activating receptors, sometimes called molecular glues and related all kinds of subtypes of the space are coming out. Rapamycin, which is the best known longevity drug, is an induced proximity drug. It pulls together 3 proteins, not just 2. It's kind of amazing, the natural products evolution that can achieve that. So just to kind of highlight, you've got a great cap table. You have Felicis, Y Combinator, H1, Jeff Dean from Google AI, Wilson Sonsini, the law firm, a bunch of prominent angels, and you also got a $3 million grant from ARPA-H recently, and some golden tickets, BMS, Astellas, AbbVie, and other pharma. So absolutely crushing it. And just speaking about, I don't know if you want to elaborate on P53 as a target. This is the guardian of the genome. It's the most studied gene in the entire genome, full stop. Fixing P53 mutations in cancer, it could apply to many different types of tumors, sort of a holy grail target for pharma. So why is it so hard to drug? It's unstructured domains, transcription factor, and how have you guys managed to get a handle on it?
Yeah, so first of all, thanks for that really nice intro. And P53 is just such a fascinating target. And it's not just an oncology target too, it's also an aging target, controls DNA repair. So we're definitely, as we look at it, we're looking at things beyond just oncology for the long run. But for the oncology aspects, so P53 is the canonical tumor suppressor in humans. As a consequence of that, it's like tautological that it has to break for cancer to form, because it's functional. Cancer cells just don't really thrive. So it's broken in almost all cancers. And generally speaking, most small molecule drugs are great at breaking things. They're usually inhibitors, like you mentioned. They're not really good at fixing things. That's why more complex therapies exist, but many of those don't really work in cancer for various reasons, like delivery. So it's just been this target where the technology didn't exist, and there's a massive amount of failure. It's a very well-known target. People failed for many decades, and kind of pharma, said we can't do it. But really, it was just the technology to drug p53 didn't exist. And so the reason it made so much sense for us, despite all the failure, is that, again, it's a transcription factor. And the way transcription factors work is that they sit in the cytosol of your cell, and some protein will come in and interact with them. Based on that interaction, the transcription factor will go to the nucleus and induce a specific transcriptional response. Any transcription factor has many different responses it can induce. So basically p53 has hundreds or thousands of interacting events that tune its function and turn on different tumor suppression programs and other things. So we thought, like, look, if that's true, if p53 is controlled by protein interactions, it's probably much easier to rescue its function in cancer with a protein interaction compared to what people tried in the past, which is just a small molecule interaction. So that's basically what our platform does. Is it says, for a target like that, can we find the right protein interaction, first of all, which is a very non-trivial problem. That's what we built all of our tech around. Once we've done that, we can make a drug that induces that interaction and then can be used. And it's a small molecule, but just much, much more powerful than like what you could get with a traditional small molecule.
Rock and roll.
Yeah, awesome. Thanks, Harborne. So last but by no means least, moving on to our final presentation, we've got Reason at Repair.
Hello. As you probably know, I'm Reason, and we're going to be talking about repair biotechnologies very quickly. We reverse atherosclerosis, and we're presently in the middle of raising a round. So as a reminder, atherosclerosis is probably the biggest problem ever. Obstructs blood vessels, kills people, a lot of people, and the therapies we have today do not get rid of this plaque, this fatty stuff that grows in your arteries. We have a therapy that does. Lipid nanoparticle mRNA therapy that in mouse studies dramatically reduces plaque very rapidly. The treated mice do a lot better. The mechanisms are a little complicated to go into in 5 minutes, but it's not just plaque regression. We dramatically improve liver health, dramatically improve metabolism, dramatically reduce a lot of problems in the body. It's a very applicable therapy to a great many conditions of aging and obesity. This past year, we had a successful FDA meeting and we obtained orphan drug designation for a rare form of atherosclerosis as our first way forward to the clinic. But let's talk about some recent updates here. Of note, recently, this is very new data that came out just from one of our long-running studies. The drug we use has a very long-lasting effect. It is, in fact, repairing a form of damage. You do the treatment, and then afterwards you get a sustained benefit. And in this case, the sustained benefit we're measuring is increase in exercise tolerance. The mice that are treated do way better on a treadmill than the mice that don't. And these are mice that started out with severe atherosclerosis. Also of note, we completed a safety and feasibility study in cynomolgus monkeys. It's safe. The human drug worked great. It produces the same changes in metabolism as in mice. This was a very promising study. We've added 2 impressive new figures to our advisory board of late. Deepak Bhatt has been a very long time investigator in cardiovascular disease, and Frederick Rahl is a very important figure in the homozygous familial hypercholesterolemia field. This is a rare form of atherosclerosis. Both of these figures are very intrigued by a way to reverse atherosclerosis. In the clinical world, this is the holy grail. If you can do this, you will attract everyone's interest, and that is what is going on as we add these key opinion leaders to our advisory board. We're in the middle of raising a Series Seed B. We've conducted a first close of $5.25 million at the end of last year, and we're seeking 11. So if you're interested, this is your chance to follow smart money and do some good in the world. Looking ahead beyond the immediate. In the very near future, the next year, we're looking at closing the Series Seed B in a couple of months, publishing a scientific paper to place our work in the context of existing work on cholesterol. We're conducting our GMP manufacturing. We'll commence a Series A roadshow sometime this year. And establishing a pharma partnership is very high on our list as well. And of course, after we have our GMP batch, we will be going into IND-enabling studies, money allowing, late this year. Looking beyond that, the HoFH indication, homozygous familial hypercholesterolemia, gives us the fastest possible path to a clinical approval and proof of concept in patients as soon as possible. And bear in mind that successful cardiovascular companies never go all the way to approval before they get acquired. They get acquired pretty much immediately on having human data is the usual outcome, and at very high valuations, because cardiovascular disease is a very expensive problem to have. Beyond just cardiovascular disease, as I said, this is an approach that can be used for many, many different forms of disease, age-related and obesity-related. The one we have data in metabolic dysfunction-associated steatohepatitis showing that we are far more effective than any of the existing drugs under development. And in the market, the one drug that's actually approved, we are very interested in neurodegenerative conditions. That includes retinal degeneration as well. And that is something we plan to work on at a preclinical stage this year as well. So it's a very, very big therapy, a very big pipeline, very many opportunities. So I'll halt there and happy to take questions. And of course, we are always interested in talking to investors who want to follow on after the smart money, as I said. And with that, thank you.
Nice. Thanks, Reason. Pretty exciting. And I think I'll say that Repair is one of our recent investments. And one of the very exciting things what we have seen is the kind of data they have generated recently. It's very rare to see a consistent and robust data across multiple models of atherosclerosis. They are notoriously difficult to generate proof of concept. So seeing something positive and similar result in non-human primates is very promising and kind of de-risk the biology here. So very interesting. Congratulations on that. And also a very positive FDA pre-IND meetings, which looks very positive. So we think like it's all set to go into clinics and hopefully we'll have a breakthrough result there. I would like to understand a bit more the pipelines you mentioned there and maybe expand more on what is the potential of this platform, cholesterol degrading platform, beyond just cardiovascular space? More elaboration on that. And the other thing, question, which I think is important to understand, based on the current standard of care, how do you see this as a patient? If I suppose I'm a patient, why would I be taking these therapies? And how does you see the combination with the standard of care, which is out there?
So ultimately, where we would like to be is you as a patient will be taking this therapy because your doctor will be telling you you're an idiot if you don't. That's the place you want to be in, which is the place you're in with statins right now, right? Your doctor will tell you you're a fool if you're not taking statins. And unfortunately, statins just don't have a very large positive effect. Ultimately, we see a world in which our therapy would be used intermittently throughout later life. You'd say once every 5 years if you're a normal human being, once every 1 year if you have a mutation that accelerates your atherosclerosis. And then in between, nobody's going to stop using statins. Everybody who can use them and PCSK9 inhibitors will continue to use them to slow down the progression of atherosclerosis to the degree that those therapies are capable of doing that. But our approach will be doing the heavy lifting of ensuring that you don't, at the end of the day, grow plaques and die. The application of this class of therapy beyond what we do with atherosclerosis is very, very broad, precisely because the excess cholesterol that accumulates in some parts of your body more than others, or everywhere if you're obese, that excess cholesterol is just a cause of harm. It is outright harmful. It's a form of damage. So every condition where you see this disruption of cholesterol metabolism leading to local excess, it happens in the brain, in the pancreas for diabetes, in the liver for liver conditions, and just everywhere. You'll find it contributing to every condition. The only question is, how big is it as a cause? And if you look at Alzheimer's, there's a whole line of thinking that is pointing towards lipid metabolism and its effects on microglia and the inflammation in that condition. And it's a very important sort of unexplored area because nobody could really get at what you need to do, which is clear out that excess cholesterol.
Very cool. I see there is a question in chat box. So does Repair Bio have SPVs for smaller angel investors? or is it only for VC large tickets? So maybe, Oisin, you want to comment on that?
We are traditionally very angel friendly, so reach out is what I'll say. I mean, obviously, we like more money rather than less money, but we come from a very small investor, angels heavy background, and we like to give small investors the ability to get into deals that they would otherwise, be excluded from.
I just want to say a huge thank you for everyone for attending, in particular to all of our founders. It's late in various parts of the world, so thank you very much for tuning in. We've seen presentations from all across the biotech and longevity ecosystems today, and there's a huge breadth of technologies and approaches for tackling health span and lifespan and extent and aging. And so hope everyone enjoyed seeing the potential. Just a reminder, if you are an investor and interested in speaking to any of the companies today, or in talking to us at LongGame, diving deeper, please do email team@longgame.vc. But for now, goodbye and live long and prosper.